today's video, we are going to be covering the muscular system. This is, again, anatomy and physiology. In order to understand some of this unit, you're going to want to have knowledge on the integumentary system, but also histology and just, interacting units like that. First thing we're going to cover is types of muscle tissues. Now, like I said, you do need to be kind of familiar with the integumentary system. So just to know, the types of muscle tissue are obviously skeletal, cardiac, and smooth. And then we should know that the only voluntary muscle tissue that there is skeletal. That's what you're moving to help you move. And the only muscle tissue that is not striated is smooth muscle tissue, which, that kind of makes sense. Smooth wouldn't have striations, whereas cardiac and skeletal would. And then in terms of location, just really quickly, skeletal, obviously attached to bone, the skeleton, cardiac, obviously the heart, and then smooth, you kind of find that in the interior lining of organs, and also endothelial tissues, the interior of blood vessels. Here's some pictures of that. Functions of the muscle tissue, we're just going to go over this pretty briefly. Obviously, they help you produce body movement. They stabilize your body position, so muscles are continually contracting to help you move, especially obviously when you're awake. And, neck muscles, for example, will hold you upright, your head upright, so you're actually able to maintain a position. Now, muscles also help with storing and moving substances within the body. For example, sphincters, which are ring-like bands, kind of rings of smooth muscles specifically, found in, let's say, the stomach, the urinary bladder, the intestines, smooth muscle within blood vessels, like I just talked about, endothelial tissue, and esophageal contractions. So things like these moving substances are done through the help of these muscles. They also produce heat, used to maintain your normal body temperature from that, shivering when you're cold, which is involuntary, and contraction also produces heat. I will insert a picture of the diagram that is supposed to go here, but we know the point of origin is going to be the attachment of the muscle's tendon to the stationary bone. So that's where the tendon, which we know attaches a muscle to a bone, that tendon is going to be specifically attached to the bone that doesn't move. The point of insertion, on the other hand, is the attachment of the tendon to the movable part of the bone. So in limbs, the origin is usually proximal, meaning like closer to the attachment point of the limb, and insertion is usually distal. So again, origin, the stationary part, is usually proximal, closer to the attachment point, and the insertion part, the tendon attached to the movable bone, is usually distal, so further away from the attachment point. And insertion is normally pulled toward the origin. So an antagonistic pair, it's pretty simple. It's just a pair of muscles working at the same joint that cause opposite movements at the joint. So literally just kind of the same idea of origin and insertion. For example, a flexor and extensor. So a flexor reduces the joint angle. Think about when you're putting your arm upwards, you're reducing that joint angle. Or when you're doing curls with weights, extensor is increasing the joint angle, and otherwise literally just putting your arm back from doing curls or something like that. Now we have anatomical organization. So the muscle in itself is really big, but it's not just like muscle. So if we go a little closer, we actually know that the muscle is made out of these groups, and these groups are called fascicles. And a fascicle is this group here, 1, 2, 3, 4, 5, but these fascicles themselves have muscle fibers, which are those smaller little muscle fibers you see in there. And that's not where it ends. Those muscle fibers are actually made up of myofibrils, and those myofibrils are composed of sarcomeres. So we're going to get into that later, but here's a diagram explaining, parts of the muscle, but here you can see the fascicles. This is one fascicle pulled out, and that one little fiber in there is just a muscle fiber. So you have the muscle, then you have the fascicles, and then you have the muscle fibers, and then as we'll continue downwards, we'll see that we have the myofibrils and the sarcomeres, which is the smallest unit of muscle. Again, this is kind of just summarizing that. Another thing to know, epimysium is connective tissue that surrounds the skeletal muscle. So here, epimysium, you kind of just have this layer of connective tissue that surrounds all of the muscle, skeletal muscle specifically. Endomysium is connective tissue that surrounds individual muscle fibers. So around this, you're going to have an endomysium. And then paramysium is connective tissue that surrounds individual fascicles. So it's kind of like a tricolon thing going on here. Epi is the very outmost, endo is the very inmost, and peri is that kind of middle layer over there. And then here is the myofibril, and the sarcomere in itself. So the sarcomere, this is made up of so many parts because it's so detailed. It's literally its own cell, so that's why, it has its own cell parts. The sarcolemma is the plasma membrane surrounding a muscle fiber, and T-tubules are tunnel-like extensions of the sarcolemma that pass through the muscle fiber side to side. So we could see the sarcolemma here, and these T-tubules kind of go all the way down. These are some things that help transport, whatever to the cell. Sarcoplasm, obviously like cytoplasm, but it's just of a muscle fiber, and it has the mitochondria that produce large amounts of ATP, which are obviously needed to contract the muscles. The reticulum, myoglobin, myofibrils, that's what I said, that's what I talked about. The myofibrils have these sarcomeres, which contain thick and thin protein filaments. So that's an important thing to note. Sarcomeres are made of thick filaments and also protein filaments. So the thin filaments are made up of actin, and you can see this on the sarcomere right here. And the thicker filaments are made up of myosin. So it's important to know the different proteins that make up the thick and thin filaments. Now, we can even break up the sarcomere structure. There's a more detailed way to talk about it. Sarcomeres, of course, compartments composed of thick and thin filaments like we just talked about, but there's also a way to kind of separate the parts of a sarcomere. The Z-discs is the dense material that separates one sarcomere from the next, so it's kind of just where you see one sarcomere starts, another one ends, so from here to here. A-band extends the entire length of the thick filaments, so the A-band right here, these are the thick filaments. It's the entire length, including even any overlap. The H-zone only has thick filaments, so this here is the H-zone, no overlap included, just thick filaments. The zone of overlap is, of course, the zone of overlap where it's overlapping. The M-line represents the middle of the sarcomere, and it kind of supports the proteins. The I-band is lighter, less dense, only the thin filaments. So from here to here, that's an I-band.